there was a lunch today with crap.
Good for
This is what this is about.
You're such a
My tree! My magnificent tree!
we should hang a swing.
Yeah, there is a good one. I mean, but Jesus, so look at all the garbage, first of all, around this tree.
Yeah, why some big
I'm disgusted. This is where I dump my garbage. Yeah, I don't use garbage. Why would someone pick a backyard? You've got a backyard. That can be where you dump your garbage. Somebody's old mice. Okay, guys. So I am the steward of this house. And the steward, literally this tree, guys, I know you can't tell by this picture, but it's this big around. Like it would take 3 or 4 of us to wrap ourselves with our arms out entirely around this tree. And it is, when I first got this house, I was like, oh, I want to update the kitchen. I want to change the siding to wood siding. I need to fix the roof. And now I can't think of anything but this tree that people have just thrown garbage around. Yeah, I am a tree-hugging hippie. It's true. But this tree is magnificent. And there's just garbage around it. And vines, you can see, I've hacked away some of the vines. There's a ridiculous shed in front of it.
Do you utilize that shed or is that not yours?
No, it is my shed. But the thing is, I want it and I want to use it, but none of it stands in the way of my tree. I'm showing you guys what I'm doing over my summer break. So, if anyone is in town and is bored and wants some exercise, you can come hang out with my garbage and help me move it away from my beautiful, magnificent tree friends. You get to hang out with me, and you get to honor a tree who is probably like 1,000 years old. Okay, you guys are just focused on the exam. I can see that. I don't know. Like, it's still so cluttered with vines and garbage. Yeah.
In my head, I always thought it was cluttered. Like, I always thought that was worse. But then, I don't know, sometimes I just why is it
I grew up swinging on my, like, I grew up in a small town. I grew up in, in the woods, playing on vines, but these are basically strangling the trees. And there's like tarp, there's tarp that's embedded in the root system. It makes me so sad. So when I get to my house every day, I just look out the window at that shed around that magnificent tree. No one's just done anything. Well, there's lots of poison ivy, so if you do want to come by and help me move stuff, you gotta be like fearless. But yeah, there's mostly just, there's lots of other trees. There's muscadine vines, so muscadine, poison ivy. But these are mostly English vines. I got a weed whacker. I got a chainsaw. This thing needs more than just that, though. It takes muscle. I mean, if this is something that, I mean, that's honestly, like, I have family, I have friends, I have people that can help, but if this is something, like, people want to, like, do, like, a little work party and set my stuff up, I'm serious, like, about honoring this tree. I really am. All right. Yeah, like, this is... I mean, this is honestly like what I plan on doing. I feel like it's going to take me my entire break unless it comes to. I got a shovel. Okay, I've always been outdoorsy, but not really like it's that kind of. It's a rounded one, but I can get whatever tool we need. Okay. But some of the stuff Oh, perfect. Oh man, it's so awesome.
What's that random fence?
Oh, that's an old door, and actually that's preventing my dogs from getting back there. All right, I've taken up too much of her time with my tree. I don't know why that's on here, but anyway. You guys want to talk about biochemistry rather than my tree? I get it. That's fine. Unless the tree is on the exam, you don't care. Oh no, I put up there because the student was asking me about beta oxidation. But I
Total isomerism. What do you want us to know in regards to that? Like, I have a question here. Which enzyme periodic cells can possibly
Yeah.
That's all we should know about that, or?
You're talking about the telomerase, right?
Yeah.
So yeah, telomerases just extend the length of the telomeres, which will extend the length of the life of the cell. Because if you can extend the telomere length, then you can extend the life of that cell, because every time it replicates, you're able to extend those telomeres. You can keep replicating that cell over and over and over. And that's just it. Like telomerase, that enzyme, its only job is to extend the length of the telomeres. But our cells turn it off, unless it's a cancer cell. Cancer cells activate that telomerase to keep those cells dividing. All of that could be a potential question, yeah. Understanding that telomerase is important for extending the length, and cancer cells utilize that enzyme in order to divide uncontrollably.
Okazaki fragments. All we have to know about that is that that's just the initial kind of section in which we have to know that Okazaki fragments and contact don't just kind of go together, or they're two separate things?
They're two separate things. So Okazaki fragments are part of DNA replication, and whereas the Tata box has to do with RNA transcription, yeah. But the Okazaki fragments, you just need to understand that you have, with DNA replication, you have a leading strand and a lagging strand.
Which is the 3'.
Yeah, because you're always going, replicating in the 5' to 3' direction. One strand is going to be continuous, that's the leading strand, you don't need to make fragments. But the other strand, the lagging strand, does it in a piecemeal kind of way, because it's making the 5' to 3' direction. It forces it to make it like just pieces at a time, and those pieces at a time are called Okazaki fragments.
But conceptually, aren't those fragments of the Tata kind of similar when it comes to the concept? Because isn't the Tata one just, when it comes to transcription, isn't it kind of the same thing that one area?
It's just, the Tata box is a piece of the promoter that's upstream of the gene. And that is, so it is, the only way they're similar in that they're both DNA sections. But Okazaki fragments are literally just made only when DNA is being replicated, whereas the Tata box, part of the promoter when you're doing RNA transcription, is always there. And that helps regulate gene expression. If the transcription factors don't bind to that Tata box, then they won't recruit RNA polymerase and you will not get gene expression. So transcription is all about making RNA in order to turn that into a polypeptide to make protein, whereas Okazaki fragments, we're just talking about DNA replication, and in that case, when you're replicating the leading strand, you're able to make a leading continuous strand versus when you're making the other strand of the DNA, a copy of that, you have to make the Okazaki fragments. And that's just only made, Okazaki fragments are only made during DNA replication. Yeah, whereas the Tata box, that promoter region, that regulation is always there.
So I understand that we combine the three. Although in regards to the difference between where that comes into play for replication and transcription.
So when you do transcription and translation, that's also in the 5' to 3' direction. So everything always happens in the 5' to 3' direction. And that just relates to the different carbons and what carbon is where.
We always have to create a 3' strand, right? Or
Nope,
we
so
don't have to.
strands, so for example, DNA has 2 strands. I should have used this, I guess. So DNA has 2 strands, and those 2 strands are the, where are you? My annoying pen, that is not an Apple pen, but you still work, so I'm still gonna use you. But I don't know where you went.
I'll talk to you about it.
Here we go. So when you have DNA replication, then essentially, remember the DNA strand is double-stranded, so you have this double helix.
Right.
And so you've got to pull this apart, and so now when you pull this apart, you're going to have 2 strands. And so one strand is going to create a new strand on this one strand with DNA replication, and the other one is as well. So both of these strands are going to be, you're going to be making a new DNA. But keep in mind these strands, so this one's 5', this strand is 5', and this is the 3' end of that one. This is the 3' because they're anti-parallel, and that's the 5' end. And so that's just referring to the ends of those DNA strands. And it always occurs, so DNA replication, when you unwind these guys, then, yeah, helicase is doing the unwinding, so helicase is coming in here, and it's gonna unwind this thing, and when it does, you're gonna have one strand that's being unwound on that side, and the other strand on this side as DNA helicase is coming in and unwinding it. But this strand, both of them are going to be made in the 5' to 3' direction. So this black one here is already in the 5' to 3', and so this is going to be the leading strand. It's going to be making a replication of itself on this leading strand, versus this other strand has to go in a piecemeal one kind of way because it's also going in the 5' to 3' direction, but it's going to be making it, again, this is the 5', 3'. No, what you're talking about now, so this is DNA replication.
Oh.
So this is just DNA replication. And you're talking about transcription and translation, which is, here's your DNA strand, again, it's in like this, but with transcription and translation, you're going to be unwinding this, but we don't worry about how it's essentially what's going to happen is not really using a helicase, but there's going to be this transcription bubble. And for example, let's say this is the gene, and that gene is going to have a promoter region that's going to be right here. And that's where the transcription factors are going to bind, and they're going to polymerate, they're going to recruit RNA polymerase II in order to make the mRNA transcript. And that's going to be the mRNA transcript that's made from one of these strands, the template strand. And so you're not using both strands when it comes to doing transcription and translation. You're just using the template strand here in order to make this one strand. You're going to copy that DNA, and that copy of DNA is going to be copied as messenger RNA. And the enzyme that's doing that is RNA polymerase II, is the one making that mRNA strand. And that TATA box is right here. It's in the promoter region upstream of that gene. So yeah, you're confusing the two different, like DNA replication is something completely separate from transcription translation, which is where you have this TATA box in the promoter region. Yes, it is a dangerous confusion. But it's good that you recognize it now, because this gives you time to think about that. But this area here, upstream of the gene, is the promoter, and that has that TATA box. That's where RNA polymerase is going to bind. And then it can transcribe that gene and make the mRNA transcript. Should we go back to my screen now, or we good? Yes.
So is it correct in saying that DNA polymerase reads 3' and 5', but synthesizes 5' to 3'?
Well, I would not try to do that. Like, I wouldn't confuse yourself in that way. Just always know that DNA bases are added in the 5' to 3' direction. So I would say, in a sense, yeah, you might be I'm not visualizing that. I could sit down with it and you're probably right. But yeah, always know nucleotides are added, whether it's mRNA or DNA replication, nucleotides are always added in the 5' to 3' direction. If you want to go back to my tree, but I'm not going to say it again. What do we want to do next? Do we want to talk about DNA replication or transcription translation again? It's mostly 60% new material. So the new material is where we started talking about nucleotides. So you have nucleotide metabolism. Was that first of the new material? And remember, this is where we make and break down purines and pyrimidines. The second part is DNA replication of the new material. And then we have RNA transcription and translation. Transcription, the fourth lecture was RNA translation, and that last part was gene expression regulation. This is all the new stuff. And this will be about 60% of the exam, so gene expression regulation is that last part. This is all the new material. Yeah. He asked a question, what were the most missed questions by the biochemistry 2 students last term? I'm going to be honest with you. You guys are not them. Like, they aced that exam 2. And I was like, they were a different energy. They were just different altogether because they had Dr. Dennison for quarter 1. And so when they had 3 for quarter 2, when I had tutoring sessions, I had to have them in here. So many students showed up. We couldn't have it in CGUS 225. And I'm just saying, like, it was, like, they were very concerned about biochemistry. And I know you guys are too, but I don't know if comparing you guys to them is a good way to do it. It really does, you guys are just not them. And it's for whatever reason, and I think it's because you guys had to be for Biochem 1, and they had Dr. Dennison, and I think that definitely just changes what you're used to and accustomed to. But it is going to be similar to what you guys are expecting, so I would focus on that, like understanding the way I teach and the way I give exams is I kind of try to focus on the big picture. I know for you it feels like details, but I try to focus on the big picture stuff. Like, what are those functions? What are the vitamins that do carboxylation reactions? What's the vitamin that does decarboxylation reactions? And I focus on that. And if you rewatch the lecture videos, I recommend that, rewatching the lecture review videos. And when you do it, and I've told you guys this before, always, because I made the review, the last term my students didn't have the review slides. But you guys have the review slides. And so when you rewatch those videos, you can sit down with the review slides and look at these questions as you're rewatching those videos. If you kind of get that concept, then that's going to serve you a lot more than revisiting exam questions. Because I thought about bringing exam questions. We can go over those true-false questions that we did, was your extra credit quiz. We can go over those if you want. But this is, if I were studying, how I would study is revisit my study guide notes. And if I had the video lectures, which I didn't have as a student, I would rewatch those and just kind of review these questions as I went, because that's going to serve you better. That's how I would study. And I would still do those practice exam quizzes that are available. I would redo those, not the weekly quizzes, but the practice exam quizzes. I'd redo those a few times, because every time you do it, different questions will pop up, and will help you establish whether or not it's helping you remember this. But don't just study the quizzes. How many times have I said that? Too many. Too many. It's true. Yeah. Which one? Nucleotide metabolism. All right. All right, I might need some help. So we'll try to do this on my own. So when it comes to nucleotide metabolism, we're focused on, so nucleotides. What are nucleotides, first of all? These guys are, nucleotides have a nitrogenous base. I gotta like stand, because if I touch, then things get messed up. Nitrogenous base, and those nitrogenous bases are your adenine, guanine, thymine, uracil, cytosine. Which one of these are purines? AG, so these are purines. And the TUC, thymine, uracil, and cytosine, these are your pyrimidines. All right, so nucleotides also mean that there is a sugar, and that sugar is a ribose. Riboses are pentoses. They're five. They have 5 carbons, and there's also phosphate groups, and they can have as many as 1 to 3. So that's why nucleotides can be AMP, adenosine monophosphate, adenosine triphosphate is a nucleotide, guanosine triphosphate, monophosphate. So depending on how many phosphate groups, that will change them to an AMP, to ATP, GTP. It could be ADP, that means there's only 2 phosphate groups attached. But these are ultimately what you're making, and these are what's going to be used to make your DNA and your RNA. As well as ATP obviously has a different function as well. This is an energy molecule, but it's also a nucleotide. It's part of your, it's going to be used to help make your DNA and RNA.
You're saying it's going to cause the energy molecules to synthesize these, right?
Yeah.
Again, when you want a particular vibration from work, sort of energy is a particular one.
So, I'm not sure. I don't know. You mean whether it's AMP or ATP? So I don't know that actually. But these nucleotides, whether they've got 1 or 3 ATP, 3 phosphate groups attached, are going to be used. Those are going to be used to make your DNA and RNA. So we're going to talk about how we synthesize them in a very basic way. I don't have all the details because you don't need to learn the details. So I don't re-memorize or relearn the details because I don't expect you to know them. But we can sit in my office if you want to know more details and talk about those. So now that you know what nucleotides are, and by the way, nucleosides, because that can be on the boards, it's on my lecture, so. But nucleosides are the same thing, except they don't have the phosphate group. So they're just the nitrogenous base, and the ribose sugar would be a nucleoside. I'll add that to my lecture at some point and test people on them. All right, so now that we know what the purines and the pyrimidines are, we're going to talk about how we synthesize them. How do we make these nucleotides? Which one of these, I'm going to use you guys today, my brain is on my tree. Which one of these purines or pyrimidines is going to start with a 2-ring structure? So purines have a 2-ring structure, pyrimidines have a 1-ring structure. So we're going to start with, we'll go with pyrimidines first. So we're going to make pyrimidines. And pyrimidines again are your thymine, uracil, and cytosine. So with pyrimidines, again, all you need to know for my exam is the starting material and the end material. There are lots of enzymes involved, lots of different there's a path. The paths are varying, but for your purposes, just understanding the starting material will help you understand how they're synthesized, because you're going to be using amino acids. So the amino acids, my memory, the amino acids that you need for pyrimidine synthesis are going to be glutamine and aspartate. I really hope I'm correct. Am I checking my work? Glutamine and aspartate, And you also are going to need a carbamoyl phosphate, which you've seen this before in what pathway? Urea cycle. So this is the starting material. You also need ATP and an extra carbon in here, but this is where your glutamine and aspartate are going to add the nitrogen groups. So the nitrogen groups are going to be coming from glutamine and aspartate, because remember, these guys are nitrogenous bases. There's lots of nitrogens in these nucleotides. Where do the nitrogens come from? The nitrogens come from amino acids. So you need these amino acids, glutamine and aspartate, to make your pyrimidines. And when you do this, you end up with your, technically you end up with UMP first, and that UMP is going to be used to make your TMP or TTP and your CTP. And all these guys, by the way, are going to need folate. More specifically, TTP or TMP more directly, but all of them ultimately will need folate, because folate is going to provide some of the carbons, the methyl group donations that are needed. But the main takeaway, they're all going to need folate to synthesize. So that is pyrimidine synthesis.
You said UMP makes TMP and CTP?
Yeah, so technically this one, UMP is made first, and that's going to be used to make your TTP or TMP and CTP. And you could write this either way, TMP or TTP. Technically, again, it's just a matter of how many phosphate groups are added. And that just depends on what your cell is going to be, what other things your cell is doing. All right, pyrimidines are also going to have to be broken down. So you have to know how to break these guys down, but it's really simple. So these guys, whether it's UMP, CTP, TTP, these nucleotides are going to be broken down. The nitrogen is going to be removed. And what's going to happen to this nitrogen? Urea. Exactly. That's it. That's the breakdown. You're going to be removing those nitrogens, and you're going to be making urea with them. Versus pyrimidine, so not pyrimidine, versus purine. So now let's do purine synthesis. So that was pyrimidine synthesis. Purine synthesis. So this one has two rings. So I'm going to try to draw the two rings here. So essentially you're going to have, it's a two-ring structure. All right, I'm just not doing a good job here. It's a two-ring structure, but it's going to start with a ribose 5-phosphate. So purine synthesis is going to require that first ring is a ribose 5-phosphate. So we're going to have to start with that. And then we're also going to add the nitrogen groups. So those nitrogen groups are going to be coming again from glutamine, aspartate, aspartate, and glycine. So this one is going to have an extra amino acid. So the starting material is different. But again, you still need nitrogen groups, and those nitrogen groups are coming from these amino acids, glutamine, aspartate, and glycine. It's going to be a series of steps, series of enzymes, but the end products are going to be AMP or ATP, and GTP or GMP. Either way, these are the nucleotides that can be used to now make DNA and RNA and other stuff, ATP. This one does have an intermediate, but I won't put this on the exam. It's an IMP, it's an intermediate that's going to be used to make those guys. So it's mostly, just remembering that starting material. And these guys have to be broken down as well. So when your purines are broken down, they're going to be broken down into something called xanthine, which is going to be converted to uric acid. Uric acid. And uric acid is what can build up. So if it's not properly excreted, then it can build up and cause gout. So gout is a result of inappropriate or dysfunctional excretion of uric acid. You're not properly getting it water soluble enough in order to get it into the urine. And so it crystallizes in your joints. And it's extremely painful and inflammatory. Yeah.
So when AMP and GMP are working, what's happening? Like, is it, they're like, just,
You guys don't like all the pictures I give you, but I can show you the pictures. I do have the pictures in my slides because I don't know those, like all the enzymes that are specific to those pathways. I'm much better at glycolysis and things like that. But we can pull up, like, that's what those pictures are for though. So when you have this, okay, I'm telling you the breakdown, or we turn them into xanthine. What is xanthine? I can't draw that structure, but anytime. Amino acids. I've seen a lot of amino acids. Okay. Oh, here we go. Wait, that's the video. Or is it? Number. So it is helpful when, I mean, I like to look at those pictures. Oh, there it is. It's right in my beginning. Because these pictures are really helpful for kind of visualizing this stuff. Because I can't draw out these pathways. I don't have them that memorized. But if we look at the pictures, you can see what's going on. And so always, so here we have, this is pyrimidines. This is the building of pyrimidines. So this is the making of UMP. That's the one that's made first. And this is the part where UMP is going to be used to make CTP and TTP or TMP. But where's the other one? And here, that's the synthesis. Degradation. Huh. Where's my degradation slide? Just kidding. Yeah, here we go. Oh no, that's synthesis. Oh, maybe I don't have the breakdown slides in here.
so
So,
like the, breakdown of pyrimidines, like the nitrogen's removed. So like, with this one, is it just like AMP or AMPAG, MP, or just like?
so uric acid is also a nitrogenous compound, and so they're going to, instead of those nitrogens being used to make urea, they're converted into uric acid.
Gotcha.
And those are the molecules that I can't really draw out because I don't know what they look like. I don't have, and I unfortunately don't have a picture of that
So
one.
it's the nitrogen that's being.
Yeah.
removed. Okay.
Yep. And that's the nitrogenous molecule is uric acid because you got to get rid of that nitrogen. But instead of urea, that's a good point. Pyrimidines are converted into urea. That's how you get rid of those nitrogens and those nucleotides. But purines, those nucleotides are going to be converted to uric acid. That's how you get rid of those nitrogens within purines.
So, it could be a question like, the pyrimidines goes to urea cycle.
Yep. Pyrimidines go through the urea cycle in order to get rid of those nitrogens, whereas purines are broken down into uric acid, and that's how we get rid of those nitrogens in purines. Yep. And that's really the big picture of purine and pyrimidine metabolism or nucleotide metabolism. I do recommend still watching the videos. I mean, I feel like I covered most of this. But, yeah, so all of them require folate, tetrahydrofolate. Yeah. All right, any other questions about nucleotide metabolism? All right, then I'll I'll take another question if someone has one on another subject. Where is one? I spit on my phone. Before I spit it on my iPad. What we could do is, I do have close without saving changes. Yeah, no, need to save that. Let's see what I have here.
Oh, good question.
Yeah.
Folate's also required for carrots.
Yes.
Yeah.
Yeah, folate is let's just say folate is needed for synthesis of all the nucleotides. Some more directly, technically TMP, the thymine nucleotide, that one's more directly, but all of them, at least even indirectly, without folate, you can't make any of the nucleotides. What I was looking for is, final exam questions. I don't know what that is. Lipid metabolism questions and answers. Oh no, what are these? Are these from the exam? This is an old exam I gave. All right, we'll do some of these. And at the end of this, we'll pick out one, and I will let you know. Like I will put one of these on the exam, and you guys will have to. But in return, you need to do the faculty. What are they called? The review. Yeah, whatever that is. That'll be like our trade. But we're gonna do this first. We're just gonna go through something. Unless you guys have questions. I know, like you like quiz questions, but I'm happy to just go through. We might come up with questions as we're doing this. So some of these might not relate to what we've done in the class, and if that's the case, we can ignore it, and I'll let you know. But this one I think is potentially fair game. So what dietary fatty acid family exerts anti-thrombotic properties? So is it omega-3 or omega-6? Omega-3, and is omega-3 alpha-linolenic? Or alpha or linoleic? Alpha-linolenic. So omega-3 is anti-thrombotic, and we didn't talk about this so much. We talked more about its anti-inflammatory properties. So omega-3 tends to be anti-inflammatory, anti-thrombotic, so it reduces blood clotting. Whereas omega-6s, like linoleic acid or arachidonic acid, these are omega-6s. They tend to be inflammatory and promote blood clotting. So remember, omega-3 and omega-6 have different properties. They both make eicosanoids, those hormone-like substances, prostaglandins, leukotrienes, thromboxanes. But the omega-3s tend to be anti-inflammatory, anti-thrombotic, meaning they reduce blood clotting, reduce inflammation, whereas omega-6s tend to be inflammatory and increase blood clotting. Both are important. You need a balance of omega-3s in your body so that you will, omega-3s and omega-6s, in order to balance those effects in your body. Sphingomyelins. Sphingomyelins are important phospholipids found in where? Myelin sheath. Which enzyme will act on cholesterol in the cell to store cholesterol? There's a D here. So hormone-sensitive lipase, lecithin cholesterol acyltransferase, cholesterol esterase, or acyl cholesterol transferase. Wow, these are. So technically, in this cell, this is LCAT, and this is ACAT. Technically, the answer is ACAT, but both of these I would have accepted if I were to put either one of these on the exam. This is probably not the best question for you guys because I was more specific with you guys, or less specific with you guys. But essentially, when you store cholesterol, you're going to be using either LCAT or ACAT. These acyl transferases are what store cholesterol. Because when you want to store cholesterol, remember cholesterol is this four-ring structure. When you want to store it, you want to put a fatty acid on it. You want to put a tail on it. And what puts fatty acids on things are acyl transferases. So acyl transferases put fatty acids on things, and when you want to store cholesterol, you'll put a fatty acid on it, and both of these will do that, LCAT and ACAT. And one of them specific for the cell, that's ACAT, and one of them specific for lipoproteins, and that's LCAT. But neither one of these, I don't care if you remember which is which, but if you do want to store cholesterol, you need one of these acyl transferases, an ACAT or an LCAT, in order to store cholesterol. That was a little. Hopefully this feels a little bit like review to some of you. All right. In the fed state, insulin stimulates lipoprotein lipase at adipose tissue. So first off, is insulin active in the fed state? Yes. So insulin is going to stimulate stuff. Is it going to stimulate lipoprotein lipase at the adipose tissue? So insulin is a storage hormone. Lipoprotein lipase is going to break down chylomicrons in the triglycerides in chylomicrons. Because remember, chylomicrons are the fed lipoproteins, so they're carrying the dietary triglycerides. And in order to get those triglycerides and to store them in the adipose tissue, you need lipoprotein lipase. So that is what's happening. Lipoprotein lipase is for storing triglycerides. The other one was hormone-sensitive lipase. This one's going to be turned off by insulin and turned on by glucagon. So hormone-sensitive lipase, this is going to be activated. So that's what you're probably confusing a little bit, Savannah. It's this glucagon is going to turn on hormone-sensitive lipase, and that is going to break down. So hormone-sensitive lipase breaks down triglycerides for fatty acid, for beta oxidation. So make sure you separate lipoprotein lipase from hormone-sensitive lipase. They're not the same thing. Lipoprotein lipase is for storing triglycerides in adipocytes. Hormone-sensitive lipase is for breaking down triglycerides, freeing those fatty acids so you can make ATP with them. This is another one you guys can do. What is the carrier for medium-chain fatty acids in circulation? Medium chain, albumin. So if it were long chain fatty acids, the answer would be chylomicrons. So chylomicrons would be long chain. But the short and medium chain can be carried by albumin. Long chain can be as well, but only when you're breaking them down. If you had a high-fat meal, ate a high-fat meal, then that's going to be carried by chylomicrons. But there are times that albumin will carry the long-chain fatty acids, but medium and short-chain, always albumin. Not the lipoproteins, it's going to be albumin. Ooh, here's one. How does that work? Match the lipoprotein with its function. So which one of these lipoproteins is going to stimulate lipoprotein lipase? ApoA1, ApoC2, or ApoE? ApoC2, exactly. ApoC2 is going to stimulate lipoprotein lipase. Let me make this bigger for you guys. Oh, you guys can't see this back there. All right, which one stimulates cholesterol storage? So in other words, it's going to activate LCAT. It's going to activate LCAT in order to store cholesterol in a lipoprotein. ApoA1, exactly. So this one is going to be ApoA1. Cholesterol storage, ApoA1 will activate LCAT so you can store cholesterol. And this is going to be in HDLs. HDLs are going to be able to store cholesterol that way. All right, so the last one then, which one is needed for cell recognition and uptake is going to be the last one is ApoE. So ApoE is going to help the liver recognize these lipoproteins. So the liver is going to be able to bring in these lipoproteins if they have an ApoE on it. All right. Sorry, what? All right. Trans unsaturated fatty acids are now considered more atherogenic, more cardiovascular problems than saturated fatty acids because they. What do trans unsaturated fatty acids do? Trans fats. It's going to be atherogenic, meaning it's going to mess with our blood vessels. And I didn't use this word, but if you've heard of atherosclerosis, it's cardiovascular disease. Arteriosclerosis is the hardening of your arteries. Atherosclerosis is when you have the plaque formation of your arteries. So atherosclerosis is a type of arteriosclerosis. But atherosclerosis, that's the plaque buildup, the clogging of your arteries. And that clogging of your arteries, we see that with trans fats. And we also know with trans fats, they elevate your LDL, which is your bad cholesterol, and decreases your HDL. That's why the food companies are being required by law to remove trans fats from foods. Because food companies tend to put them in foods. But they're not allowed to do that anymore. All right, the fatty acid structure below is an omega blank fatty acid called. So I bet if I count all this, and I don't want to count all this. So this is not one you would have on the exam. Yeah, no, this is not one I would put on the exam. I'm just going down the lane, so. All right, so this next one, and these aren't in order. I don't know where this came from, to be honest. I can't remember why I have this. Why might someone who is drunk have high blood lactate levels? You guys don't need to know this either. Nutrition, I used to talk about alcohol as well and how that affects the system. You can actually get good at drinking alcohol. You can increase enzymes. So why is someone a lightweight with alcohol and why someone's not? Not on your exam, but you can actually increase enzymes in the liver to help you break down alcohol. So if you want to get good at drinking alcohol, you just have to practice. Not recommending it. Okay.
Does meat have the same effects?
No. That's something, yeah. That's more of a neuro effect versus alcohol also has a neuro effect, but in relation to enzymes and how we break it down. So I don't know. I don't know. Well, no, that's interesting. I actually don't know much about marijuana breakdown. I mean, we store that one, so we don't break it down as much. Yeah, we're getting off topic anyway, but I don't know. That might be one I might have to look up at some point. So all this stuff on alcohol, I'm just removing. A lot on alcohol. Oh, Oregon, I miss it. For so many reasons. No. I mean, these are good questions, but these are like for upper level nutrition students. Like you guys would kill me if I put these on the exam. Upper level nutrition? No, I don't think you guys will. You guys don't have to do in like a lot of this.
Why are you teaching
Because they're pulling me into biochemistry, guys. They won't let me teach more than like 2 major classes at a time, and until they find someone else to teach biochemistry, which I'm more than happy if they do. But I mean, other than the fact that I like meeting the new students coming in, I don't like these questions. I'm gonna try to find another one. Close without saving juice.
There's so much to work.
I have to be careful because some of these might be exam questions. I got grades in here. I have to be really careful what I actually click on. I'm gonna get in trouble. Yeah, do you guys have any questions while I kind of look around in here? Do we want one of these questions that I just had? Do we want to look at my CV? How do I have my CV in here? I have no idea.
I actually have a really quick question.
Yeah.
Are we gonna have to know all of the DNA polymerases?
No, you only need to know DNA polymerase 1 and DNA polymerase 3. You don't need to know all the eukaryotic DNA polymerases. Huh? I don't know about 2. I don't know what 2 might do.
So one cuts off 3 extends, right?
No, well, DNA polymerase 3 grows the DNA chain. DNA polymerase 1 removes RNA primer and replaces it with DNA. So close. Good thing he's not teaching is what I want to say.
There's 3 RNA polymerases.
3 RNA polymerase, yeah. Yes. Oh, I didn't hear that part, but yeah, they do tRNA. Yes. Did I just do this one? Oh, this one's got the answers. No, we don't want the answers. All right, this might be a good one. All right, but I wish the answers weren't on here. Let me find one without the answers. Without CVG. All right, that's his question. I might try that one. Maybe this one doesn't have the answers. Darn it, they all have the answers. All right, so this one must have been from one of your quizzes. Yeah, this is just one of your quizzes questions that I accumulated. Yeah. Yeah, so remember the DNA polymerases, there's a lot of eukaryotic ones, but we learn DNA replication via the prokaryotic because it's a simpler form. If we try to learn eukaryotic, that's for upper level biochemistry students. Yeah, you're welcome. But yeah, that's why they are prokaryotic, yes.
So that's why I was just seeing DNA polymerase 2 then, right? Or am I completely off?
You're thinking of RNA polymerase 2, I think.
Okay.
There's RNA polymerase 1, 2, and 3, and there's DNA polymerase 1 and 3.
Okay.
All right. I'm looking for a question for you guys. So, yeah, in what form is most body fat stored? Triglycerides. Triglycerides with saturated fatty acids of long chain length tend to be what at room temperature? Solid. So the liquids would be the unsaturated fatty acid. Yeah, so you need to know the RNA polymerases.
Is there an extra credit exam?
I don't know what you mean. Alright, I feel like we're like at that, like, you know, what do we have a question outside of, like, on the material? Do you guys wanna talk about the RNA polymerases?
Sure.
So there's 3 RNA polymerases. RNA polymerase 1 that does a few things, but in this case, it mostly makes the ribosomal RNA. RNA polymerase 2, as soon as I just try to relax, it won't let me, makes the messenger RNA, so that's the important one. That's the RNA transcript. And so this is RNA polymerase 1, this is RNA polymerase 2, and RNA polymerase 3 makes what? What's the only mRNA or RNA that's left? tRNA. Yeah, RNA polymerases are just transcription translation, DNA replication, that's DNA polymerase. That's the only one we only 2. DNA polymerase 3 is the one that replicates the DNA, makes the growing DNA chain, and DNA polymerase 1 is the one that removes the RNA primer and replaces it with DNA once that chain is almost finished. Sorry? DNA polymerase 1 is the one that removes the RNA primer and replaces it with DNA.
So which one places it?
What puts the RNA primer on the DNA?
Primates. Cholesterol serves as a precursor for all the following except, so if we make our sex hormones, bile acids, vitamin D, these are all coming from cholesterol, but phospholipids, we do not make phospholipids from cholesterol. I wish this didn't have the answers. This would be a lot more fun. So, but this is actually one of your quizzes. So these come from like